Glycolysis is the first step in cell respiration. It occurs in the cytoplasm, and all the reactions in glycolysis are catalyzed by a specific enzyme. Energy is first made available in the first step of glycolysis when the two ATP molecules are hydrolyzed. Next, at the end of one glucose molecule, the first ATP molecule attaches to one end of the glucose molecule in the second step of glycolysis. Following that, the glucose 6-phosphate, which is also known as G6P, becomes rearranged into an isomer known as fructose 6-phosphate. The second ATP molecule attaches to a phosphate group at the other end of the fructose 6-phosphate. This forms fructose 1,6-bisphosphate. Because of this, the ATPs transfer a phosphate group to the backbone of a 6-carbon. The backbone is then split apart and forms glyceraldehyde 3-phosphate, which is also commonly known as G3P, and dihydroxyacetone phosphate, which you can also hear it as DHAP. The DHAP is then converted into G3P. Step 6 of glycolysis has two parts. In the first step, each of the G3P give up one hydrogen and 2 electrons to NAD+, which reduces to form nicotinamide adenine dinucleotide. In the second part of step 6, the G3P will combine with inorganic phosphate, resulting in 1,3-bisphosphoglycerate, which is an unstable molecule.
Oh, no. This can't go here.
Why?
In step 7, a phosphate group is readily given up to ADP by 1,3-bisphosphoglycerate, which forms ATP. Now, because of this, 2 ATP molecules are created, which is called the substrate level. The 8th step includes 3 phosphoglycerate molecules rearranged to a structurally different isomer known as 2-phosphoglycerate. The second last step of glycolysis includes a new molecule, 2-phosphoglycerate, giving up 1 proton and 1 hydroxide ion, which forms water. This results in a phosphophenylpyruvate, also known as PEP. The final and 10th step of glycolysis is when PEP breaks down because it is very unstable. This releases enough energy to transfer one phosphate group to ADP, which results in another ATP molecule.
So next up we have pyruvate oxidation, which is the second stage in cell respiration. Pyruvate molecules enter the matrix of the mitochondrion with the help of membrane carriers, which are also known as transport proteins. They pass through the outer and the inner mitochondrial membranes. Coenzyme A attaches to the acetyl group of the pyruvate molecule, which enables it to be oxidized, while NAD+ is reduced and becomes NADH. As you can see in the picture above, you can see the NAD+ becoming the NADH. The remaining carboxyl group is removed as waste in the form of CO2, as seen in the image. And in the end, the resulting product is acetyl-CoA, and that is a picture of the molecular diagram of acetyl-CoA. Next up is the Krebs cycle, which is also known as the citric acid cycle. So in the Krebs cycle, it is the third stage in cellular respiration. The Krebs cycle is a cyclical series of 8 reactions in order to transfer energy, to form organic molecules into usable forms of the cell. So examples of this are ATP, NADH, and FADH2. And at the same time, CO2 is released as a waste product. So the Krebs cycle occurs in the matrix of the mitochondrion and in glycolysis. Each reaction is catalyzed by a specific enzyme for that reaction. So as you can see in the picture, the matrix is the middle of the mitochondrion. So step 1 of the Krebs cycle. It begins with acetyl-CoA from pyruvate oxidation reacting with oxaloacetate to form citrate. The CoA is then removed. So citrate undergoes an isomerization to form isocitrate. And the picture does not show isomerization, so it is written in. In step 3, through a redox reaction, isocitrate is oxidized to become alpha-ketoglutarate, and an NAD+ ion from the matrix is reduced. That is then gaining electrons to form an energy coenzyme, which is NADH. So before, CO2 is also released as waste, and then alpha ketoglutarate forms succinyl-CoA by binding with CoA and being oxidized by NAD+, which is then reduced to NADH, while CO2 is then removed. In this step, succinyl-CoA loses its CoA to become succinate, but not before some phosphorylations take place. So a phosphate from the matrix will displace CoA, and GDP is phosphorylated to become GTP. As you can see in the picture, ADP is then created to become ATP when the phosphate group is transferred to the ADP. Another ion from the matrix, FAD, oxidizes succinate, which then forms the energy coenzyme FADH2, and then succinate becomes fumarate. In step 7, water is added to fumarate to form malate. In step 8, which is the last step, malate undergoes one more redox reaction with NAD+ to produce NADH and oxoalicetate, and after that, the cycle begins again. So overall, pyruvate oxidation produces 2 molecules of NADH. The Krebs cycle produces 6 NADH, 2 ATP, 2 FADH2, and at the end of the Krebs, glucose is completely dismantled. So the next step that comes up in cellular respiration is ETC.
ETC is found in the intermitochondrial membrane, and there are 7 major components of ETC. ETC's job is to make ATP. The steps of ETC are: the first step in the electron transport chain is when the proteins in ETC are set up in order of the increasing electronegativity. So the first component is the weak electron acceptor, and the last component is the strong electron component. The second step of ETC is when the electrons are passed from one component to the next along the membrane. So the NADH binds to the NADH dehydrogenase, which is the first component. Then the FADH2 binds to succinate dehydrogenase, which is the second component. So because of this, hydrogen ions are pumped into the intermembrane space. FADH2 works the same, but it starts at the succinate dehydrogenase complex. So because of this, electrons from the FADH2 pass through fewer electron carriers, and therefore less ATP is made. Each molecule of NADH produces 3 ATP and 3 hydrogen ions, while each molecule of FADH2 makes 2 ATP and 2 hydrogen ions. When the 2 electrons have left the cytochrome oxidase, they combine with oxygen and hydrogen to form water. In the end, the goal of ETC is to create an electrochemical gradient in order to power ATP synthesis. The end.